with that molecule is a regenerative therapy for osteoarthritis, which could be combined with anti-inflammatories. We're looking at spinal muscular atrophy, spinal muscular atrophy is our other people. And the whole idea here is to compensate for the mutation in SMN1 by altering the splicing or upregulating SMN2. Most people are looking at splicing, modulating splicing like ISIS, okay, and Roche. We found a molecule again with a phenotypic cell-based screen that simply upregulates gene expression. So it should be actually synergistic with splicing modulators. And again, we've done chemistry on these molecules, and we just put them into PK/PD models into the delta 7 SMN mouse, and we see really nice PD responses with upregulation of SMN levels in the transgenic mouse model. So we're working really hard here to get to a clinical candidate, and we're quite optimistic we'll be there reasonably soon. We're also looking at tissue-targeted drugs that aren't systemic. So we've focused on the GI tract, we've focused on lung, we're focused on eye. And so one area we're looking at is ENaC inhibitors for cystic fibrosis, COPD, and dry eye. So ENaC is clearly a target for rehydrating the lung, which you really need to do in COPD or CF, or even dry eye. ENaC's the receptor in the eye. The problem with most drugs that have been made that do this is they're systemic. So you also have ENaC in the kidney, and so you give this drug, you rehydrate the lung, but you get hyperkalemia because you're also hitting ENaC in the kidney. So that's a problem. So if you just make a molecule that stays in the lung and it's lung localized, you should overcome that issue. It's a really simple idea. So we have a really, a nice platform to do that across a lot of disease areas, and we've done that, okay? And interestingly, the molecules you make are 100 times more potent in the guinea pig TPD model in terms of activity than the best compounds in the literature, okay? But they're not more potent on the channel. It's because they're localized in the lung and you get massive exposure, okay? We can't measure the systemic levels of this drug. So we're not going to have hyperkalemia because the drug just doesn't get there, and it doesn't look like metabolites do either, okay? So the beauty is that we have this, and right now we're actually going to take it into the clinic in dry eye, because again you have ENaC in the eye. It turns out these molecules, because they're tissue targeted, are incredibly persistent. So we think we'll have great persistence in the eye, okay, and we'll rehydrate the eye, and these molecules will not go anyplace else, okay? So we hope to do those studies. We hope to actually initiate that clinical trial this year independently, and at the same time we're taking these into the sheep model for COPD, and we have 2 different molecules, so we may partner those in different ways. As I said, we are applying this platform and this approach to elastase inhibitors and a lot of other tissue-targeted drugs. We've gotten into immunotherapy, both in the antibody-drug conjugate space, and here we're looking at antibody-drug conjugates not only for cancer, but also for NASH by targeting T3 to the liver, okay? And inflammatory disease, targeting PDE4 inhibitors, okay, to macrophages, for example. Okay, so you overcome the safety issue with that molecule simply by targeting it. So, and we're also looking at interesting bispecifics with new technologies, and we've gotten into CAR-Ts as well. And again, there's so many players in this space. What could